Hello and welcome back to Smart Science Tutorials. You go to destination for making complex scientific concepts easy and fun. I am Jagruti and in today's video we will be giving into the fascinating world of inverted microscopes, a critical tool in biological research. Whether you're a student or a budding scientist, this video will help you understand how this unique microscope works and why it's so important in studying living cells, tissues, and other delicate specimens. Have you ever wondered how scientists study cells in petri dishes or observe living organisms without disturbing them? Well, the inverted microscope is the answer. Unlike traditional upright microscopes, an inverted microscope has its objective lenses and light source placed below the stage, making it ideal for studying samples in liquid cultures or dish. It's an essential tool in fields like cell biology, microbiology, and medical research. So in this video, we will take you through the structure and components of an inverted microscope, show you how to use it and explore its various applications. So if you're curious about how this incredible instrument works, keep watching. Don't forget to hit that subscribe button and turn on the notification bell so you never miss out our educational videos. Let's get started. Introduction to inverted microscopes. Begin by explaining what an inverted microscope is and why it is different from the standard upright microscope. So, an inverted microscope is designed with the optical components inverted or flipped compared to a regular upright microscope. In an upright microscope, the objective lenses are above the stage and the light source is beneath the stage. In an inverted microscope, the objective lenses and light source are below the stage and the specimen is placed on the top of the stage. So, why use an inverted microscope? So, uh, because uh, we can observe large or liquid-based specimens. In upright microscope, specimens are usually placed on glass slides. However, some specimens like living cell cultures are often kept in petri dishes or culture flasks. Because we observe large or liquid-based specimens, in an upright microscope, specimens are usually placed on glass slides. However, some specimens like living cell cultures are often kept in petri dish and culture flasks containing liquid media. And upright microscope setup would require moving or manipulating this large sample, which can be disruptive. With an inverted microscope. The specimen is placed on the top of the stage, which means it's easy to view liquid-based or large specimens without disturbing them. As well as we can view samples from below. The inverted configuration allows the microscope to focus on specimens within the container. Example petri dish, eliminating the need to transfer cells to slides or use complicated mounting techniques. This makes the inverted microscope ideal for studying live unmounted samples. Main components of inverted microscope. The light source. The light source located beneath the stage. The light source can be adjusted for intensity brightness depending on the needs of the experiment. So LED lamps or halogen bulbs are typically used and modern inverted microscope allow for precise control for the of the liquid brightness, which is especially important for delicate living samples. Condenser focuses light onto the specimen from beneath the stage. The condenser is responsible for gathering and directing light towards the specimen. The light passes through the condenser illuminates the specimen and then travel upward to the objective lens. The condenser has an adjustable aperture, which is diaphragm, that controls the amount of light passing through, which helps optimize image contrast and resolution. Stage, where the specimen is placed. In inverted microscope, the stage is horizontal and the specimen sits above the stage, typically in petri dish, culture flask, or multi-well plates. This configuration is especially helpful when working with liquid cultures, as it allows scientists to examine the specimen without needing to transfer it to a slide. Then the objective. In an inverted microscope, the objective lenses are located below the stage rather than above it like in a traditional microscope. These lenses come in different magnifications. Example 4x, 10x, 20x, 40x, and 100x. The user selects the appropriate objective lens to observe the specimen clearly. So the objective lenses are fixed and the stage moves up or down to focus on the specimen rather than the lenses themselves. Eyepiece. The eyepiece is where the observer looks to view the magnified image of the specimen. Some inverted microscopes are equipped with cameras or digital screens for observation, which is useful for recording images or sharing with others. Next, how to use an inverted microscope. Once the basic components are understood, how to use the inverted microscope? This is a step-by-step process. First, prepare the sample. Place the specimen in a petri dish, culture flask, or multi-well plate. Ensure that the specimen is adequately prepared. Example cells in growth medium, live organisms in water. Second one, adjust the light source. Turn on the light source and adjust the intensity using the controls provided. This is important for both bright field and phase contrast microscopy. Third one, select the objective lens. Start with a low magnification lens, 4x or 10x, to get a general overview of the sample. If higher magnification is needed, rotate the nose face to switch to a higher objective, example 40x or 100x. Focus the image. Use the coarse focus to bring the stage up and down slowly, adjusting the focus so the image becomes clearer. Use the fine focus to move small adjustments for sharper details. Adjust the condenser. Fine tune the condenser to optimize the light depending on the technique. Example, bright field phase contrast. Next, observe the sample. Once the image in the focus, observe the specimen and make any necessary adjustments to the illumination or magnification. Types of illumination used with inverted microscope. Inverted, inverted microscopes can be used with various illumination techniques depending on the sample and the research question. First one is bright field microscope. So this is the most basic form of illumination where the light passes directly through the specimen. It's used for stained specimens or those with natural contrast. It produces a bright background and dark specimens. Second one is phase contrast microscopy. Phase contrast microscopy enhances the contrast of transparent unstained specimens, such as living cells. It uses differences in the phase of light as it passes through different parts of the specimen. This allows you to see structures like cell membranes, organelles, subcellular structures without needing to stain the sample. Third one is differential interference contrast, DIC. DIC is similar to phase contrast but uses a more complex optical setup to create high contrast 3D images of specimens. It's useful for observing fine details in live, unstained cells or tissues. Fluorescence microscopy. Fourth one is fluorescence microscopy. Inverted microscopes can be equipped with fluorescence modules that allow the observation of specific proteins, cells, or tissues that have been labeled with fluorescent dyes. This technique is used to study things like protein localization in cells, DNA interactions, or microbial infections. Applications of inverted microscopes. After covering the theoretical and practical aspect of inverted microscope, so we are discussing the different applications and why it is so valuable in various scientific fields. So first one is cell and tissue culture. Inverted microscopes are crucial for observing live cell cultures in a dish or flask. Researchers can observe cell growth, division, migration, and response to various stimuli. without disturbing the sample. Second is live cell imaging. Since the sample remains in its natural environment, inverted microscopes are used to track live cell behavior over time, making them ideal for studies on cell division, migration, and response to drugs. Third one is microbiology. Microbial cultures grown in liquid media or on solid agar plates can be observed easily. This is important for studying the behavior of bacteria, yeast, algae, and protozoa. Medical and diagnostic research. Inverted microscopes can also be used in medical research, particularly for observing tissue samples, studying pathogens, or diagnosing diseases in live cell cultures. To summarize, an inverted microscope is a versatile and essential tool in biological and medical research. It allows researchers to view large or liquid based specimens, such as cultured cells, without disturbing them. The key features include the placement of objective lenses beneath the stage, various illumination methods such as phase contrast and fluorescence, and the ability to observe live cells in their natural environment. By understanding the structure, use, and applications of the inverted microscope, students will be able to better appreciate its role in modern scientific research and its importance in fields like cell biology, microbiology, and medical diagnostics.